So in this session, we will try to understand genetic engineering, how it is applied for crop improvement. Though the crop improvement has been tried with traditional methods like mutation breeding for ages, and it has been successful. But the new technology which was developed in last decade or two, it has an advantage. The advantage is that it is more specific and precise, whereas mutation breeding is random and it requires a lot of efforts in terms of selecting the right kind of change in a plant we are looking for. Whereas the newer technology, like genetic engineering, is more precise, wherein we try to identify the genes which are responsible for a particular trait, and then use that to very specifically alter a plant for that trait, using that gene. In codes, we call that as a technology. So the question is why we want to, what we want to change and why we want to change in a crop plant. The plant productivity, when we talk, is dependent on or is both qualitative and quantitative. When we talk of quantitative improvement, we are basically trying to improve the yield of a crop per hectare. And when we are talking about qualitative improvement of a crop plant, we are referring to the quality of the produce in terms of nutritional values, in terms of newer product which can be made in a plant, And nowadays, plants also have been used or made to produce what we call as biofactories, wherein a plant can be made to make things which normally it doesn't produce. For example, vaccine production. It means we can transfer a gene from humans into plant where it will produce that protein, or what we call vaccine, and that will lead to the production of edible vaccines. So with this broad introduction, we'll try to see what are the areas wherein this genetic engineering can be used. So as I mentioned that this can be for qualitative and quantitative. So quantitative production of a crop plant depends on biotic factors and abiotic factors. Biotic factors include the factors which influence a plant productivity, like infections from bacteria, fungus, viruses. These are the major factors which influence plant productivity in the field. And also, the plants are eaten or infested by insects, pests. So that is another factor which affects the quantitative plant productivity in field. Similarly, abiotic stresses like salinity, soil salinity, water, drought, flooding, herbicides or weeds, for example, I mean, weeds are the plants which grow in the field along with the crop plants. So that reduces the crop productivity because they then uses the resources which a farmer gives to the plants. And then quantitative, qualitative, qualitative improvement. For example, we can change the shelf life of a plant. For example, a tomato successfully has been demonstrated, and I will talk about this later, that how the shelf life of tomatoes has been increased. What it means that the time from field to uh, the consumer is increased. You know. So, and then nutritional quality is changed. For example, a hot example is the golden rice, where normally rice doesn't produce vitamin A, but then now it is possible that the vitamin A can be produced in rice. Similarly, a plant can be made to produce pharmaceutical like vaccines in the industrial product like polymer. Normally, these chemicals are produced in the uh, in the in the factories, but now it is possible to make polymers in plants, and then it can be extracted out from plants. So these are some of the applications of this technology of genetic engineering for crop improvement. Now, when it comes to what all it requires, uh, um, to develop this technology, actually. So if we, these are 5, 6, 7 steps which one needs to go through in order to develop a technology. For example, you have to identify a set of gene or genes which are responsible for that trait or characteristic one is talking about. There is a whole lot of technology involved in that, which we will be talking about in due course of the in future lectures. Okay, so the first very important aspect of technology development is identifying the genes which are responsible for a particular trait, or in other words, linking trait with genes, or what we call functional genomics. After having a gene or set of genes responsible for a trait, one should have a technique or technology optimized for gene transfer. Now, these genes need to be transferred into plant cells or callus or tissues. Okay, so there are a set of technologies which have been developed and are in use. We will talk about these technologies later. Uh, but then one should have the gene transfer technology in hand, and then the plant regeneration protocols to regenerate a plant from the transformed either chloroplast, callus, or tissues to get a transgenic plant. And then once you have the plants in hand, you need to check whether the transgenic plant has the gene of interest or transgene has got integrated, and then it is expressing in the right quantities, which will alter the trait, or which is required to affect the trait phenotypically. Uh, so all this needs to be checked before we really confirm that a plant is transformed or undergoing genetic change. And once that is confirmed, then these plants are used in the normal breeding programs via back cross, repeated back crosses, they are then transferred to the varieties which we want to transform, or which we want to really take the character in. Okay, so once you have stabilized the trade, then they go to field testing. You know, field testing for the trade which one has modified. You know, and once that is confirmed, then it has to go through approvals. Each country has its own set of rules before this genetically modified organism or variety is released for commercial users. So, to conclude, what I'm trying to discuss here is that there are a set of steps which are needed to be followed before a technology is come to the use. Okay. Now we'll go one by one, actually. So when it comes to identifying genes, so there are a whole lot of methods which have been tried for for identifying and isolating genes and then characterizing them for the function. This is the first step in the technology development. So to name a few, if you see the gene discovery involves, there are two approaches. One is the hybridization based techniques and another is the PCR based techniques. So in hybridization based techniques, you have you make the cDNA libraries, and then these libraries are differentially screened. What is this differential screening? Is you screen the same library 2 times, one with the probes from normal plants, and then the second round is done by the probe generated from the test conditions. And you pick up the genes which are expressing only in the test conditions, and then you go ahead with that for further characterizing that. So that is one, and then you have subtractive hybridization. So this is where you try to subtract. You have 2 sets of plants. So you have RNAs from control conditions and the test conditions. You you mix the both and then try to pick up only those RNAs or mRNAs which are in the test conditions, and then clone those things and generate the set of genes which are expressing only in test conditions. And then you have microarray DNA chips, oligo-based and the EST-based. This is very effective approach to pin down set of genes which express only in test conditions and not in normal conditions. Then if you come to PCR-based techniques, you have DDRT-PCR or APRT-PCR or RAPD-RT-PCR. Again, the concept is the same, that you try to pick up set of genes which are expressing only in test conditions. The difference here is that you take out the polyMRNA and then go make cDNA from there, and then use different sets of primers to amplify and see that you get an amplification only in test conditions and not in control. And very specifically, you isolate that and then clone it and then go for sequencing. So these are the set of techniques which have been used in use for zero down set of genes or gene which expresses itself only under test conditions and not in the control conditions. Once you have a gene in hand which you have cloned, by using any one of those approaches, then what needs to be done is that you need to have the sequencing done and then complete the full length gene sequence. You know, that's very, very important. And once you have this sequence, uh, you can use that sequence as a probe and then try to determine the copy number of that genome, and then expression pattern by using another analysis, or nowadays real-time PCR is the another approach by which you can determine that the gene which you have picked up is expressing only in the test conditions and not in the control conditions. You know. And then once you determine that a set of gene or the particular gene of interest is expressing only in test conditions, then you go for functional analysis. And that functional analysis is done by transgenic approach. That is, you try to either overexpress this gene or you silence that gene in transgenic plants. You know, so if the gene is involved in a function, and if you are silencing it, then you will lose that trait in the transgenic plants. You know, and if you are overexpressing the gene, then and if it is involved in a particular trait, then it is going to protect or it will strengthen the trait, you know, where it is involved. Once that is done, one can also look at the promoter analysis by doing promoter reporter fusion approach to make sure that the gene in hand is responding to a particular stimulus, you know. And lastly, you have to determine the in vivo cellular localization and where this protein is localized within the cell, organelle, where it is located in the nucleus, mitochondria, chloroplast, or cytosolic protein, whether it is associated with the cell membrane or where it is localized. So that's also a very important aspect in gene characterizations. And then finally, you have to find out the functions of the proteins, what it does in the cell. That is the final step for assigning the function to the protein that can be in vitro. So, so these are the steps which are involved in identifying and characterizing a gene or linking the gene to a function. Now, when we talk of gene transfer methods, actually there have been various approaches which have been used, and all of them have been classified into three main categories. You have biological methods, you have physical methods, and then you have chemical methods. And all of them have been used successfully to transform a plant cell. But out of this whole lot of list, there are two methods which have been used, include the agrobacterium-mediated gene transfer and the particle bombardment systems. They are in use these days. We have already discussed in greater detail what is this system of agrobacterium gene-mediated gene transfer. So here you take the tissue from the recipient plant and then infect these pieces or tissues with agrobacterium and then put for selection for the transgene integration on a selection medium. And this selection is done on antibiotic presence of antibiotics. And so all the cells which are antibiotic resistant are then regenerated into whole plant. So this is how, in short, the agrobacterium mediated gene transfer works. Similarly, the particle bombardment system uses a machine, what we call gene gun. So here the genes are cloned into plasmid, which has the coding region as well as promoter and a terminator. And then this plasmid is coded on gold particles or tungsten particles, and they are bombarded onto on the tissues, cells, of plants, and then after bombardment, they are put for selection, and then you regenerate plant in presence of antibiotics as the selectable marker. So both these approaches have been successfully used for transformation. Now, as I mentioned at the beginning, these are the applications of those. You generate transgenic plants for resistance against biotic stresses, where you have insect resistance, virus resistance, and the fungal and bacterial resistance. So these are the factors which affect the plant productivity, in field, because crop plants are infested by insects, different kinds of insects, and then you have viruses, fungus, bacteria, which causes different kind of diseases. So, and which once it is, once any of these factors infect the field, it reduces the productivity, and sometimes they result into almost complete loss of a crop. They are so serious factors. So it is very, very important that we have crops which are resistant to these factors, so that we can increase the productivity. Similarly, you have resistance against abiotic stresses, which includes, as I told you, weeds. Against weeds or high salt concentration in the soil, droughts, flooding, temperatures, high and low temperatures. Because again, these are the factors which influences the plant productivity in the field. And then we can also apply transgenic biology for improvement of for crop yield and quality. For example, extending shelf life of fruits, as I mentioned, like tomato, for example, and there are others where it has been successfully applied. And then you have improved nutrition contents of a crop, like golden rice, for example, I gave. Or we can change the oil contents of a oil-producing crop like sunflower, mustard, or the quality of oil, how much, what kind of oil a plant will produce. Even that can be changed, and there are examples where it has been shown. And then this technology has been used for making plants as biofactories, that is like edible vaccines. So you can make the vaccine in a plant, edible part of the plant, like fruits, like banana, for example, potato, or leaves, which are eaten, like spinach, for example, or tomatoes, which are eaten as salad materials for it. And then you have, as I mentioned earlier, polymer production. So there are whole lot of lists which has been successfully demonstrated using transgenic biology. So we have reports where genetic engineering has been successfully utilized for uh, generating transgenic plants against to tolerate high salt concentrations, which can withstand high drought or flooding and high and low temperatures, and also herbicide. We will be talking about these success stories as we go along in future lectures. You know, and then, for example, vaccine production. If you see. Uh, This is not something new. It is about 3-decade-old concept. People started working on this for the problems which people were facing in the traditional vaccines, like the uh, transportation. It requires very specific conditions. The traditional vaccines, they require uh, very expensive storage conditions and the transportation from one place to another. And then the painful pricking, for example, are required to administer the vaccine. So when the transgenic biology came into or became a success story, then people thought that why not we make these vaccines in edible parts of the plant so that they can grown in the field like any other crop plant, and then then when it is eaten, it is easy to transport from one place to another and also easy to administer. And we will have a detailed lecture on this aspect as we go along. But then to conclude here, this story, uh, uh, we are not able to we are not able to release any such edible vaccines. It's all in lab and trials, different kinds of trials. Similarly, insect resistance, one of the most popular application or production of this insect resistant plants is using BT technology. Now, what is this BT technology? There will be a detailed lecture on this. Uh, Actually, BT is the bacteria, what we call Bacillus thuringiensis, which has insecticidal property. So this is being used by farmers as a solution, BT solution in the field to control insects. So as the understanding grew on this, that what kills the insects of this bacteria, now we understand that these bacteria, they basically synthesize a protein called BT toxin or BT toxin protein, or which are designated as try 1, 2, 3, 4, 5, like this. And these endotoxins are very specific to specific insects, and therefore a whole lot of studies have gone in to link a specific cry protein to a specific type of insect. For example, cry1 is very effective or kills like butterflies and moths. Cry2 kills butterflies and flies, and Cry3 kills beetles, and Cry4 kills only flies. So these kind of understanding has been developed, and the first success story in this came wherein the BT technology was used in cotton by a seed company like Monsanto. And in India, the only transgenic or GMO which is released commercially is the BT cotton. So we will have a detailed discussion on this subsequently. Now, if you see, rice is the staple food for for 60–70% of the world population. And the rice as a whole has undergone its own has undergone a lot of improvement in terms of qualitative production and the quantitative production in the field, using mutation breeding programs, and also a large number of technologies have been developed to protect rice from both abiotic and biotic factors so that its productivity can be increased. And we'll talk about this in future slides. But then here, the point I'm trying to make is the production or generation of golden rice. Now, this golden rice actually is the basically it produces vitamin A along with other proteins and the carbohydrates. Now, why it was thought basically to have people who are who come from poorer strata of the society to supplement vitamin A in their diets through rice only, because we have globally large population which cannot afford vitamin A rich diet. And vitamin A is very, very important component, particularly for pregnant women during the pregnancy to support the growing child in the womb, and also after the childbirth, it is an important component of the diet. So this has been a very, very remarkable discovery as far as transgenic biology is concerned. And now the good news is that this crop is going to be in field, because this has been distributed free of cost to various countries, and where it has been retrogressed to the local varieties. And now we will have the local rice varieties making vitamin A in them. So on shelf, you will see very soon the sale of these kind of rice product. And as I mentioned earlier, that we have products available, particularly in tomato, where using antisense technology, the one gene was identified which is responsible for ripening, for fruit ripening. Now, if you decrease the expression of that gene by using antisense technology, then this process of fruit ripening is delayed. The advantage of this delayed ripening is that when the farmer gets time from field to taken to the market, and in market it goes to the consumers. Otherwise, tomato, once it starts ripening, it ripens very fast, and sometimes when it reaches to the market, it is already rotten. And so plants as biofactories, for example, I mentioned biopolymer production successfully. This polymer has been used in plants, polybeta hydroxybutyrate, or PHB. This was produced. It means that the metabolic process responsible for this, all the genes which are responsible for the production of this chemical, is cloned and transferred to Arabidopsis and or mustard plant or any other plant, wherein this was made by the plant, and then it is extracted from these plants. So then, instead of making it in the factories, they are made in plants and then extracted from there. So it is very, very safe that way also. And then there are some applications which are very, very interesting, actually. For example, you are going to have very soon the tearless onion. Uh, uh, the gene which is responsible for synthesizing those pungent uh, metabolites in onion, those genes have been silenced, so that you have an onion, when you cut, you won't have the tears problem. And then you have products like colorful cauliflower, for example. So you can have extracts in market, cauliflower of different colors. You have green, violet, and orange color, cauliflower. But if when you produce these kind of products, they look very sort of fancy, but then acceptability, whether socially, whether people will be easily eating the orange cauliflower, because psychologically we are tuned for eating pale cauliflower or whitish pale cauliflower, not the orange or violet and green. Green is okay, but broccoli is there. But then, even if you have these products, the point I'm trying to make is the social acceptance, for a product which one is generating. And similarly, you have colorful cones. Uh, it is possible to make a cone colorful like this or like this. So again, the point here is whether one would like to eat or take in hand and eat these kind of products. So, to conclude this session, what I have tried to do is that how the genetic engineering has revolutionized the field of agriculture, and these success stories, which I have tried to summarize, there are many actually, but then the success stories which I have put here uh, encourages one to, it's a matter of thinking something and you can achieve that. you know you you just identify a trait and link that trait to a gene or set of genes, and then move those genes into the plants and see that you have a desired product in hand. So with this sort of introduction, I will now move on to take you very specific examples how that is done. See, that is very, very important to know that how a plant is made which can withstand higher biotic stresses or abiotic stresses, or how a plant can be made a vaccine, and then what are the problems associated with it, even if you have a product in hand. As I mentioned, that the governments globally are discouraging use of genetically modified plants. For example, in India, only example of GMO which is being commercialized is the BT cotton. Although we have so many products in pipeline, if you remember, we had this BT brinjal story, which was stopped. It was almost ready to be released in India, but then it is stopped at the government level. But having said this, globally, there are large number of crop plants, genetically modified crop plants, which are being grown. For example, in US, there is at least you name a crop and they are there. In China, for example, there are they are growing all sorts of genetically modified crops and using them without any issue. So I end this session with to meet again.